This video tutorial is about the light-dependent stage of photosynthesis. The light-dependent stage of photosynthesis occurs in the grana, or the thylakoids, of chloroplasts and involves the photosystems. It involves the direct use of light energy. This stage consists of light harvesting at the photosystems, photolysis of water, photophosphorylation, which is the production of ATP in the presence of light, as well as the formation of reduced NADP. Oxygen, the byproduct of photosynthesis, is also produced in the light-dependent stage. Now, as I've mentioned before, there are two types of photosystems. I described these briefly in a previous tutorial and outlined their main roles. But just to summarize again, in photosystem 1, the pigment at the primary reaction center is a type of chlorophyll A, which has a peak absorbance of red light of wavelength 700 nanometers. In comparison, in photosystem 2, the pigment at the primary reaction center is also a type of chlorophyll A, but this has a peak absorbance of red light of wavelength 680. In photosystem 2, there is an enzyme that, in the presence of light, splits water molecules into protons, so hydrogen ions, electrons, and oxygen. The splitting of water in this way is called photolysis. Some of the oxygen produced during photolysis is used by the plant cells for aerobic respiration. But during periods of high light intensity, the rate of photosynthesis is greater than the rate of respiration in the plant. So much of the oxygen byproduct will diffuse out of the leaves through stomata into the surrounding atmosphere. Water is the source of protons, so hydrogen ions, that will be used in photophosphorylation. Water donates electrons to chlorophyll to replace those lost when light strikes chlorophyll. Water is the source of the byproduct oxygen, and water also keeps plant cells turgid, enabling them to function. We're now going to look at photophosphorylation. Photophosphorylation is the generation of ATP from ADP and an inorganic phosphate in the presence of light. There are two types of photophosphorylation. Non-cyclic phosphorylation, which involves photosystems 1 and 2. It produces ATP, oxygen, and reduced NADP. And we also have cyclic photophosphorylation, which only involves photosystem 1. It produces ATP, but in smaller quantities that are made by, or smaller quantities than that are made by non-cyclic photophosphorylation. Both non-cyclic photophosphorylation and cyclic photophosphorylation involve iron-containing proteins embedded in the thylakoid membranes that accept and donate electrons and form an electron transport system. Now I'm going to go through what happens in non-cyclic photophosphorylation. When a photon of light strikes photosystem 2, its energy is channeled to the primary pigment reaction center. The light energy excites a pair of electrons inside the chlorophyll molecule. The energized electrons escape from the chlorophyll molecule and are captured by an electron carrier, which is a protein with iron at its centre and is embedded in the thylakoid membrane. These electrons are replaced by electrons derived from photolysis. When this iron combines with an electron, it becomes reduced, so Fe2+. It can then donate the electron, becoming reoxidized, Fe3+, to the next electron carrier in the chain. As electrons are passed along a chain of electron carriers embedded in the thylakoid membrane, at each step some energy associated with the electrons is released. This energy is used to pump protons across the thylakoid membrane into the thylakoid space. Eventually, the electrons are captured by another molecule of chlorophyll A, in photosystem 1. These electrons replace those lost from photosystem 1 due to excitation by light energy. A protein iron sulfur complex called ferredoxin accepts the electrons from photosystem 1 and passes them to NADP in the stroma. As protons accumulate in the thylakoid space, a proton gradient forms across the membrane. Protons diffuse down their concentration gradient through special channels in the membrane associated with ATP synthase enzymes. And as they do so, the flow of protons causes ADP and inorganic phosphate to join to form ATP. As the protons pass through the channel, they are accepted, along with electrons, by NADP, which becomes reduced. The reduction of NADP is catalyzed by the enzyme NADP reductase. The light energy has been converted into chemical energy in the form of ATP by photophosphorylation. ATP and reduced NADP are now in the stroma ready for the light-independent stage of photosynthesis. Now to look at cyclic photophosphorylation. This uses only photosystem 1. As light strikes photosystem 1, a pair of electrons in the chlorophyll molecule at the reaction center gain energy and become excited. They escape from the chlorophyll and pass to an electron carrier system, and then pass back to photosystem 1. During the passage of electrons along the electron carriers, a small amount of ATP is generated. However, no photolysis of water occurs, so no protons or oxygen are produced. No reduced NADP is generated. Chloroplasts in guard cells contain only photosystem 1. They produce only ATP, which actively brings potassium ions into the cell, lowering the water potential so that water follows by osmosis. This causes the guard cells to swell and opens the stoma. Finally, I'm just going to show you a diagram that summarizes the main events of cyclic and non-cyclic photophosphorylation in a special diagram known as the Z scheme.